bit, but first it's more important, depending on what your starting material is, you might not have the choice. So if we click through the next slide, a couple of things you should consider is both your downstream analysis and your starting material. So, yep, you can click one more time. Perfect. So cells are recommended for things like site-seq or TCR and BCR sequencing, RNA splice analysis, but they're actually not recommended for things like attack-seq. And then for starting material, material that's been flash frozen or like large or fragile cell types using cells is not recommended there. However, if you're enriching a sample for a specific cell type, we actually aren't quite there yet for nuclei, so you should stick with cells. And then Allie, if you click a couple times, some areas where you actually have a choice include 3' RNA sequencing, which is great because it's the most common, and when you're starting with fresh tissue or slowly frozen tissue, so something, for example, frozen with a cryoprotectant like DMSO. So next slide, what are those strengths and weaknesses? So dissociation for single cell sequencing does tend to take longer than nuclei prep, about an hour, versus nuclei prep can be done in as little as 5 minutes, which I'll talk about in a little bit. And it's more difficult to preserve gene expression when you're using cells. However, single cell sequencing does have a quantitative QC. So you can just get viability data by flow, for example. And when you're working with nuclei, that QC is going to be done via just a microscope. And so it's totally qualitative. And I have an example of that in a later slide. And then single cell sequencing can give you higher complexity than nuclei. And as far as cell type composition, single cell sequencing can be more biased against fragile cells, and nuclei-seq can be more biased against immune cells. So if we go to the next slide, there is actually a paper that was published in Nature in 2020. I was about to say last year, but it's 2022 now. So a group was actually comparing single cell and single nucleus sequencing from matching samples for different tumor types. And what they saw was that the immune cells were more prevalent in cell preps, and parenchymal cells were more prevalent in nuclei preps. So they were running single cell and single nuclei sequencing. You can see the cell composition mapping in this figure just to show the differences in cell types. And this is just reiterating that we're still learning the best techniques and when to apply them. So really just wanted to highlight the importance of knowing what the biases will be when you're analyzing your data. So if we go to the next slide, let's say you know you want to work with nuclei. What would be the best method? So this is our nuclei extraction workflow. You can start with up to 200 mg of fresh or frozen tissue in this case, since you can work with frozen tissue for nuclei. And you would put it in a pre-chilled C-tube with our new nuclei extraction buffer. And you'd lyse on the GentleMax Octo, which is it's just a 5-minute program for tissue lysis and homogenization. It's really fast. And you can process up to 8 samples at a time on the machine. And then you would wash and filter your nuclei suspension. You could also further clean it by sorting it on the Tyto, and then move on to your single nucleus analysis. So since the nuclei extraction buffer is a new product, we do have some data here to share here. So this could be used for virtually any tissue. It includes snap frozen, also OCT embedded or fresh tissues. And the graph on the left is just showing you yields for both the snap frozen and fresh mouse brain and tumor tissue. And then the chart on the right is showing RNA integrity number, or RIN values, for RNA extracted from those nuclei suspensions. So this buffer has been validated for RNA analysis. It's a really great option for nuclei extraction. And then in the next slide, this slide just really is pointing out a couple of things. One is kind of what you're looking for that QC of nuclei under a microscope. So the nuclei here are outlined in pink. And what we're looking for is nice round intact nuclei with no blebbing. But one other thing that it points out is the difference in your starting material and what type of sample you get from them. So here on the left, we see this is a mouse liver sample. These have only been filtered, so no other cleanup steps. They're pretty clean. And then if you look on the right, this is an adult mouse brain sample. Again, it's just been filtered, no additional cleanup. And you can see this definitely is going to need a further cleanup step. So not all your starting materials are going to be created equal. So make sure you're optimizing your protocols depending on what you start with. So at Miltenyi, we have different workflow solutions for helping you achieve a sample that meets all of those requirements I mentioned in the first slide. So when you're working with solid tissue, this often starts with proper tissue storage. So you can hit the next slide. As we know, it's often not possible to process your tissue right away. Maybe you have a situation even where the tissue is actually being shipped to you before you process. So for those situations, we have our tissue storage solution. And this is used to maintain cell viability and cell composition. And other options include long-term storage like slow freezing or snap freezing, as I mentioned, that will only allow you to recover nuclei. But then if we go to the next slide, our tissue storage solution has been validated to prevent necrosis, prevents apoptosis, helps you maintain your cellular composition, and it helps you maintain your gene expression pattern, which is really important for sequencing, right? So to demonstrate this, we have mouse tumors here that were dissociated and then either immediately after dissection fresh or after storage in the tissue storage solution for up to 48 hours. And we looked at viability via flow, which you can see on the left, no substantial differences there between fresh and frozen, or stored, excuse me. And on the right, we also look at gene expression in fresh versus stored tissue, and we were looking specifically at stress and toxicity pathways, and we didn't see any upregulation of stress genes after those 24 or 48 hours. So the tissue storage solution is a great way to kind of extend your fresh tissue kind of life. And on the next slide, you can click through so the data shows. This is actually a paper that was published in 2021 in Genome Medicine, and they were actually showing the potential for viably frozen tissue for single cell sequencing. A lot of people actually don't think you can do any frozen tissue for single cell sequencing, but it is possible. So they slowly froze small tissue chunks like 1 to 2 millimeters in a media containing DMSO. And by slow, I mean 1 degree per minute down to minus 80, so really, really slow. And they did perform a dead cell removal step to get their viability up above 80%. And I'll talk a little bit more about dead cell removal later. But what they saw was that they were able to actually conserve the tumor heterogeneity in those frozen cells and tissues, which is nice. So it is an option as long as you are viably freezing your tissue samples. So the next step then in the workflow would be the dissociation, right, of your tissue into your single cell suspension. Here you have a couple options. You can dissociate with enzymes at 37 degrees. You could also use a cold adapted enzyme, which is becoming a little more popular. I'll talk about that in a little bit. Or you could go without enzymes, right, and just rely purely on mechanical force. So each method has some pros and cons that have already kind of been discussed in literature, which we won't go over, but cold adapted protocols are gaining a little more interest lately to reduce the risk of gene expression alterations. And we at Miltenyi have focused in the past on optimizing an enzymatic dissociation of tissues at 37 degrees, since we have shown that it gives the highest viability and yield while preserving the cell composition. So Miltenyi has the GenOmax technology for this dissociation. It combines mechanical disruption with enzymatic disruption, digestion. So that enables both the processing time and the amount of force to be minimized. And it gives you the fastest, most gentle treatment of your cells possible. So for example, on this slide, some protocols do use solely enzyme digestion, and this tends to be less efficient when compared with the GenOmax. So here, this is actually customer data of a comparison of a manual protocol for lamina-propria digestion with the respective GenOmax protocol, and you can see there's actually a much higher yield of Tregs when you use the GenOmax. And the reason for this is that sole enzymes aren't going to be able to get into those areas within the tissue, and it actually makes it less effective and it elongates your incubation time. So the GenOmax is constantly disrupting your tissue, so the enzymes are really effectively getting into all the areas of the tissue. And on the next slide, other protocols may opt for the opposite, right? So they may do mechanical dissociation and no enzymatic digestion. So we demonstrated this here by dissociating tumors, either with or without the enzymes in our tumor dissociation kit. And by using the kit, we actually are able to increase our yield of activated T cells by over 3-fold. So on the next slide, just to kind of sum up the benefits of the GenOmax, it's really allowing for fast and gentle dissociation. And the really great thing is it's fully automated walk-away fashion. You put your samples on the machine, you walk away, really reduces hands-on time, and it really actually decreases variability as a result of tissue processing. And again, you can process up to 8 samples independently on this machine. So you don't have to wait and batch all your tissues together before you start processing. And just to kind of further enable that reproducibility, we do have over 20 tissue-specific dissociation kits. These kits have really pure enzymes, known activity levels, really lot-to-lot consistent. And for a few of our kits, we do have preserved epitope lists, which can be important for experiments that use feature barcoding like SiteSeq. And as mentioned before, we do have, for tissues that are snap frozen or cells can't be isolated, we have nuclei extraction buffer. So on the next slide, I'm actually going to talk a bit about the warm versus cold dissociation. So typical enzymes that are used for dissociation, they work best at 37 degrees. But recently, a cold active protease has been gaining interest in the single cell sequencing community. So this is an experiment from a group in Australia that is comparing warm and cold dissociation of mouse kidney for single cell sequencing. And so if you look at the warm dissociation protocol, they're quartering a mouse kidney and they're using our multi-tissue dissociation kit on the GentleMax. It's about a 45-minute protocol with heat, and then they filter, wash, filter again before sequencing. And then if you look at the cold dissociation method, they're going to be mincing that kidney into a paste on ice using a scalpel. They then put it in the GentleMax C-tube with their cold active protease, and then they combine that with titration on the GentleMax in a cold room. It's about a 40-minute protocol before filtering, washing, filtering again. So right away, I want to point out that this protocol uses the GentleMax for dissociation, but the cold adapted protocol is much more hands-on. They're having to mince that kidney into a paste, and then they're also combining titration, right? So this right away indicates that the cold protease might not be as effective as a traditional enzyme. And then in the paper, actually, the authors don't comment on the total cell yield from warm versus cold, but it's our experience that effective enzymes are really important to attain good cell yield. So we're going to look at the data now. So on the next slide, we can see that a longer protocol might potentially, and potentially lower cell yield might be a fair trade-off if you're able to maintain your tissues at 4 degrees to inhibit transcription during dissociation. So the authors looked here at 17 different stress response genes, and what they found was that expression was higher in the cells that went through warm dissociation compared to cold. They also looked and saw that the stress response does vary by cell type. So some cell types showed a significant upregulation of stress genes during the warm dissociation, and other cell types actually showed no transcriptional response. So that's really important for you guys to consider when you're looking at your single cell sequencing experiments. So on the next slide, I just have kind of a summary of the differences between warm and cold. The biggest advantage of warm dissociation is that we know that it works, right? So we have validated protocols for a wide variety of tissue types, and we know that it produces high yields of viable cells. So when you're looking at a dissociation method, you really should consider how much time and how many resources you're willing to commit to optimizing a new protocol, and how important is it to have a standardized protocol that can easily be reproduced in other labs. However, dissociation-induced ex vivo gene alterations are a big factor to consider. So you should think about how transcriptionally responsive to stress your cells of interest are. As we saw in that paper, some cells had no significant dissociation-induced alterations, while others had significant alterations. So some groups actually have turned to using transcriptional inhibitors during warm dissociation just to prevent expression alterations, and you can find examples of that in the literature. And then finally, it has been shown that there are differences in cell type composition between warm and cold, but there's not really enough evidence to make any general conclusions on that yet. So on the next step of the workflow then would be dissociated samples now need to be cleaned. So you can click through one more time. We're going to want to remove any unwanted material like cell clumps, debris, sometimes red blood cells. And the same is true when you're working with blood samples. And as we noted before, for nuclei suspension, sample cleaning can be performed via flow. But first, I'm going to show you some additional methods of cleaning up your cell suspension. So if you're only going to do bare minimum for sample cleaning, that would be filtering your cell suspension. It's really important to get large debris and cell aggregates out that can clog the microfluidics of single cell sequencing instruments. So for larger volumes, we have the smart strainers, and for smaller volumes, we have the preset filters. Both of these are stackable, which is really nice. You can do a serial filtration in one step, and they come in 3 different mesh sizes each. And then we also have our debris removal solution, which is a ready-to-use density gradient reagent. And we have, depending on what your sample is and whether RBC lysis is necessary, we have an RBC lysis solution, which only requires a 2-minute incubation time. And then arguably, if we go to the next slide, one of the most important steps in sample cleanup is going to be maximizing viability. So why is viability so important for single cell sequencing? Dead cells can create two problems. They can leak cellular RNA into the solution, and this can increase background noise, can also contaminate other cells with non-endogenous transcripts. And second, dead cells still contain RNA, which can make functional libraries. So when you have a lot of dead cells in your suspension, it becomes difficult to distinguish live from dead cells. So our dead cell removal kit is a fast, easy way to help increase viability of your sample. And the kit shown here consists of magnetic microbeads. These bind to dead cells based on the annexin 5 dead cell marker, and the cell suspension then gets passed through a max column, sits in a magnet. The dead cells are retained in the column, and the live cells flow through. The whole process is about 25 minutes, and it's really gentle. And on the next slide, I just wanted to demonstrate with these tools in hand, can we obtain better data from single cell genomics, right? So do they work, and does the processing time lead to any detrimental effects on the data? So we have a collaborative study here with 10x Genomics where we analyze tumor samples after different cleanup steps. So condition A, cleanup is just minimal filtering with a 70 micrometer smart strainer for those large aggregates and debris. Condition B, we add on an RBC lysis step. And then in C, we add that dead cell removal kit that I just talked about. And in some cases, we actually found a single round of dead cell removal didn't give us the desired viability that we wanted. So we added a second round as condition D. And then we assessed the metrics on several performance metrics like, on the next slide, cell recovery. So cell recovery actually yielded the most striking differences between each cleanup condition. When you look across each tumor type and replicate, the number of cells recovered increases with additional cleanup steps. And in some samples, you can see that it increases very dramatically, like from 500 to 1,000 cells with minimal cleanup to hitting the target of 5,000 cells after the full cleanup workflow. And on the next slide, just to kind of demonstrate the effects of poor cell recovery, this can have a really significant impact on the resulting data. So the T-SNE plot on the left is from a mouse melanoma tumor sample that was only filtered, so condition A. And the plots on the right are from samples that get the complete cleanup workflow, including that dead cell removal. And what you can see here is it's really increasing your sensitivity and reproducibility. And you're also really getting a better picture of the complexity of your sample. So you really want to optimize your cell recovery by performing all of those cleanup steps. And so on the next slide, hopefully I have convinced you it's really important to have high viability, but we know that our dead cell removal kit at Miltenyi, it's not your only option. So how do you know which one would be best? And luckily, a recent publication actually compared several dead cell removal methods. They used frozen PBMCs for this study, so it's really common to get reduced viability after cryopreservation. And they tested 4 methods for dead cell removal here. So one is a low spin wash, and then our Miltenyi dead cell removal kit, which is the column-based cleanup in the pink, and a competitor dead cell removal kit, and lastly, a density-based cleanup. And both the low spin wash and the density-based cleanup are relying on the fact that dead cells tend to be less dense than live cells. So in the graph on the left, Miltenyi's dead cell removal kit is actually the only method that gives significantly improved viability. And then an important thing to note on the graph on the right is the white bars here are showing the fraction of cells that you lose after each method. So any kind of dead cell removal method that you perform is going to result in cell loss. So it's really important for you then to consider the most important thing for your project. Is it increasing viability, or is it having as many cells as possible? And depending on what you're looking for, you might want to choose something with a little less cell loss, like a low spin wash, for example. So then on the next slide, once your sample is clean, you might want to enrich for your cell type of interest in order to maximize the number of target cells that you're actually sequencing per sample. So let's just take a look at how our MAX technology enables a gentle cell separation. So if we go to the next slide, and you can just click through, so all 3 steps are showing. These are the 3 easy steps for cell separation. And the first is magnetic labeling. So this is where an antibody, which is conjugated to a very tiny superparamagnetic microbead, binds to a receptor or a protein on the cell surface. And then in the next step, the cells are passed through a column positioned within a magnet. And then the cells that are labeled with the microbead get retained in the column, and everything else is washed through in a really gentle way. And in the elution step, step 3, the column is then removed from the magnet, and that enables the magnetically labeled cells or the positive fraction to be washed out. And this separates your cells into a negative and a positive fraction with really exceptional purity and high viability since it's so gentle. So let's see what we can do with this in the context of single cell sequencing. So this is another study we performed with 10x Genomics, and it looks at single cell gene expression analysis of isolated neurons from adult mouse brains. So these are brains from 6-week-old mice. We dissociated using the GentleMax Octo with heaters and our adult brain dissociation kit. And that kit includes a red blood cell lysis and a debris removal step. So finally, neurons are then isolated using our neuron isolation kit. And then single cell gene expression analysis was performed on a 10x Chromium platform, both with cells prior and after isolation of neurons. So after dissociation, you can click through, Ali, just to show this whole slide. You can see that there are actually only 5% of neurons in the cell suspension, as that flow analysis shows, that red box. And single gene expression analysis is actually giving us 23 distinct cell populations, as you can see on the UMAP plot on the left. So this includes microglia, astrocytes, like vascular cells, immune cells. And if you look at that table, we're looking at yellow immature and mature neurons. If you count that up, it's only 2% of your data set. But then if you go to the next slide, after isolation, the neurons were enriched now to over 95% of the population. And this is reflected in the gene expression analysis. So the plot on the left is showing a majority of the cell population is immature neurons, over 80%, and a minor population are mature neurons, about 2.5%. And all those other neural cell types are successfully depleted from the sample, including microglia and those immune cells, astrocytes, oligodendrocytes. And so isolating your neurons upstream of sequencing is actually allowing you a more sensitive analysis of those neuronal cell populations. And it really gives you a lot more bang for your buck when it comes to sequencing data. And we know that sequencing can be expensive, so that's really important. And if we go to the next slide, this is not the only way to isolate your target cells. So there is another way that Ali is going to talk about, which is the MaxQuant Tito.
Awesome. Thank you, Christine. Okay, so let's move on to our second option of isolating specific cell types, right? And we're super excited because we also have a Tito clearly on site at David's core. So all the stuff that we're going to talk about now, you guys can potentially actually see for yourself when using it. So let's go and kind of give you a little background on the Tito. Okay, so droplet sorters are common, right? And, there's a lot of droplet sorters in all the flow cores, but some of the downside effects of these sorters is that they do charge and decompress the cells and apply, a good amount of pressure. And that can lead to some stressful conditions. So an alternative solution is a microfluidic sorter like the MaxQuant Tito. And this instrument works with a single-use cartridge, which you see on the right there. On the bottom of the cartridge is a chip that contains a valve. If a fluorescently labeled cell of interest passes the lasers, the valve's going to open and direct the cell into the positive collection chamber. And then your non-target cells will be collected in the negative collection chamber. And you can do multiple sequential sorts without compromising cell availability. And really the benefits, especially in, talking about how it relates to single cell sequencing, right? And the kind of comparison. So traditional droplet cell sorter, you have a high volume of sorted product, and it definitely requires a centrifugation step post-sort to prepare for 10x. Also, sometimes they can be laborious to set up and maintain, depending on your expertise with the system. But with the Tito, the sorting actually creates a concentrated product in whatever buffer you want to use, which can often be put directly into sequencing. So that's really one of the highlights of this instrument in regards to single cell sequencing, is that it actually concentrates your sample, and you can also sort in the buffer of your choice. And then, especially now with our upgrade to our auto mode, our Tito 1.0 software version, the sort setup is truly minimal, and users with little to no cytometry expertise can operate the instrument, and the cleaning of the instrument is simply throwing away the cartridge. So I know, especially for University of Chicago, when we installed the instrument, I think it was March of 2020. So we actually weren't able to really go on site and train people, but we were able to do this all virtually. So it just kind of really emphasizes the ease of use aspect of the Taito as it pertains to operating the sort yourself. Okay, and here's a little bit more in-depth picture of the Max Quant cartridge, right? Because this cartridge is the actual heart of the Taito system. Like I mentioned before, it has the 3 chambers, the input, the positive, and the negative. All chambers can be accessed from above through lure locks, either just using long or gel loaded by pet tips. And then different from conventional droplet cell sorters, the Taito has no fluidic paths inside the instrument. In other words, the cells are actually not running through the instrument itself. Instead, prior to the sort, the cells are just loaded into the cartridge and remain in the cartridge also after the sort. So you have complete access to your entire sample. You will never lose your sample. Everything is kept within the cartridge. And then we can zoom in here on the microchip just to kind of show its size for scale, right? That's the size of the microchip, but that's really the heart where the valve is located as it opens and closes like a door as it sees the cells of interest pass by. So that's just a little bit of an introduction to kind of how the system actually works. But I think what's really important is kind of the reviews, testimonials, and also applications in which we've been successful upstream of single cell sequencing. So I'm going to pass it along to Carter to kind of go into more of the technical aspect of these applications. So Carter, go ahead.
Thank you, Allie. So we're going to talk about a few of the applications, as Allie mentioned. So the first one we're going to go over is sorting of these mirroring endothelial cells prior to NGS. So the overall summary was they took these 10-week-old mice, isolated the mammary gland, got some single cells, and then we isolated these epithelial cells based on EpCAM and CD49F expression. So libraries were created, but the main thing that was done is there was a head-to-head comparison of the Taito versus the Max Quant. So on the next slide, we're going to summarize some of the data as we kind of did this sorting head-to-head on these epithelial cells. So the next slide shows that both the Taito and the Max Quant did show the same kind of 3 major cell populations. These basal, the luminal 1, the luminal 2. So we did have similar results in identifying these 3 major populations. However, on the next slide, you can see that we did capture a higher number of genes on the far left side. The Taito captured more genes per cell and a higher UMI, or unique molecular identifier. So this is showing that the Taito might have had a little higher quality than the FACS-based system due to that really, low pressure, gentle sorting. On the next slide, you can see that this really is reinforced by these stress responses on the Taito sorted cells versus the traditional FACS-based sorted. So you can see that the Taito had less stress-induced response, and you can on these different markers as listed on the right. So the Taito just showing that it really is a gentle compared to the FACS-based system. And on the next slide, instead of generating a library, they actually performed that same head-to-head comparison, but instead cultured these cells. So they cultured those endothelial cells after sorting, and you can see the Taito sorted cells did form a greater number of spheres as opposed to the FACS-based system. So it just shows that the cells sorted on the Taito in that gentle sort did have increased functionality after being sorted as well, as represented by the enhanced spheres. So the next slide. We're going to get into a different application of sorting nuclei. So this just shows the traditional FACS-based system applies charges, has high pressure, has decompression, has that big deep dive. However, on the Taito, it's more like a lazy river. We're kind of just flowing through nice and gentle. So they did sort some nuclei on the Taito. So showing the next slide, we have our input versus our sort fraction, or our positive fraction. So you can see the input, it was a lot dirtier. We were able to identify nuclei via expression of 7-AAT and DAPI simultaneously. So looking at the sorted cells, you can see just visually how much cleaner it is, how much more pure those nuclei are. So we are able to identify and sort those small nuclei on the Taito in a very gentle manner. Let's go to the next slide. So this is just a testimonial on the ease of use. It's saying that they did have great success in sorting nuclei from multiple tissue types on the Tyto for both human and mouse, and it was user-friendly with these low pressures. So you can always see that there are some testimonials saying that it does do a great job at sorting these nuclei. If we go to the next slide. This was done at the University of Chicago. So this was done with the Wilson lab, but essentially they were looking at some B cells and the antigen-specific B cells in terms of the SARS-CoV-2. So the next slide shows the overall workflow. Initially, the patients did need a PCR positive test, so they did collect leukocytes from patients that had been exposed to COVID-19. Then they isolated PBMCs, did a pan B cell enrichment, and then they stained these B cells with CD19, a few other common B cell markers, and then also the COVID-19 antigen probe. So that was the main one. And then we sorted these cells on the MaxQuant Tito. So the next slide shows that it was a really rare population of antigen-specific cells coming into the Tito, less than 1%. On the far left side, you see, about 0.26% were positive for this antigen. So we were able to sort out that low frequency of cells on the Tito, and then this team generated a UMAP library just showing the different populations with the 10x and monoclonal production based on the spike protein, the open reading frame 8, open reading frame 7a, and then they did a couple of different things like ELISA, LA-spot, and different assays. But if we go to the next slide, you can see that they did identify these 12 B cells clusters, your naive, your innate-like, and your different memory B cells post-sorting on the Tito. So just really affirms that you can sort that low frequency population of cells very gently on the Tito and generate kind of those libraries after the fact reliably. Let's go to the next slide. And here is the actual testimonial, just saying that they did use the MaxQuant Tito to sort these B cells from patients with COVID-19. And it was very quick and simple. And since the Tito is the cartridge or that closed system, you can sort without minimal risk or hazard to the operator. And it does give a high cell viability. So let's go to the next slide. This is an example of identifying two biomarkers in human oligodendrocyte progenitor cells. So the next slide shows the overview, but the main thing was that due to the fragile nature of these cells, they wanted to use the Tito because the Tito is nice and gentle, it doesn't apply charges, and it has a low pressure system. So you can see that initially they cultured these fetal brain stem cells, differentiated them into these oligodendrocytes, and they sorted them on the MaxQuant Tito. So they sorted according to the NG2 and A2B5 markers, and then did some assays downstream. So if we go to the next slide, we can see that these cells that they were nice, they're kind of fragile cells, but they were able to sort them successfully on the Tito with minimal loss in viability. And they did different assays downstream looking at cell migration and showing that the A2B5 negative and the NG2 negative did have a higher percent cell migration. And they also had higher cell proliferation based on those OD values. And then these cells were transplanted and they did show myelination abilities in the actual mice. So these are all cells sorted on the Tito. And in summary, they were able to sort these HOPCs and obtain high number of sterile, high purity cells with a high viability, just showing that the Tito really is good for those really fragile cells because it's gentle to the cells, doesn't apply charges, and it is sterile. Yeah.
Awesome. Thanks, Card, for going over those applications. I'm just going to go through a couple more kind of testimonials to kind of show you some perspectives and what people have done. Here's another biocompare review saying the sorted cells possess high purity, viability, and functionality, highly recommended. Again, all things that were emphasized by Christine that are super important for improved data quality for 10x. And then the system is perfect for sorting specific populations. And since the sample sorts into a negative chamber, if any issue arises, it's easy to resort the sample with no sample loss. This is, again, emphasizing more so of the user experience with the Tito. But again, with that single-use cartridge and everything being kept in the cartridge itself, it's really easy to adapt. In summary, the Tito microchip sorting technology is really a component of these 4 pillars and really what kind of differentiates us in the market. So it's sterile and it's safe since it does not generate aerosols. It's also an extremely used system now that we especially have our upgraded Taito 1.0, and it does not require necessarily a dedicated trained operator. I know Carter has trained users virtually and helped them get started on the Max Quant Taito at University of Chicago. You also obviously have the support of the great workers there to help as well. There are no drop delays, no calibration steps. You don't have to worry about that. Flow control, laser alignment, and sort timing are automatic and self-regulating. So that kind of emphasizes more on the ease of use. And again, like I mentioned, the sterility with it being a single-use cartridge, you just throw the cartridge out, and that's how you clean it. So let's go and just briefly, now that we've kind of covered the reasons and methods that, of how you could isolate your cells, let's go back to kind of the sample prep workflow and look at the last step before moving on to library prep, right? So the last step before loading your cells onto the Times Chromium or another single cell sequencing platform is to get an accurate cell count and to adjust to the proper concentration. So on most single cell sequencing platforms, the input cell concentration will determine your cell recovery. So overestimating the cell concentration will decrease your cell recovery, whereas underestimating could lead to an unacceptably high cell multiplet rate, right? So we can spend a whole hour on cell counting best practices and comparing different methods. But I'm just going to address a couple of key points, right? So number one, try to clean up your sample of debris and cell aggregates the best you can, as they can cause inaccurate counts, especially on automated counters relying on Trypan Blue. After kind of leaving the cells undisturbed in the tube for 10 minutes, the concentration at the bottom of the tube will be much greater than near the top. And to ensure you really get an accurate representation of the population, mix the cell suspension well before drawing a sample and taking an average of 3 counts. These are just some of the best practices we recommend. But really, the MaxQuant analyzer, which is our flow cytometer, is unlike many other flow cytometers in that it actually relies on volumetric pipetting. And this can do an automatic and absolute cell count every single time you run a sample, right? So the cell concentration is automatically provided in the analysis without the need for beads or complicated methods. You don't need extra reagents. Every single time you acquire a sample, you can actually generate that cell count. And additionally, we can get a number of other QC parameters from a single analysis. We can assess the level of debris in our sample. We can measure the cell multiplet rate by looking at the forward and side scatter properties. And we can also assess viability quickly by staining the cells with PI, right? So that is just another technology that we offer to kind of work into the entire workflow. So now we have officially covered every step in the sample prep workflow upstream of your single cell analysis. And we can kind of just go over a couple of the quick points that we hope you guys walked away with. Going back to kind of Christine's presentation, cell preparation is a critical step for successful single cell genomics analysis. Really, the method can influence your recovery rate, performance, reproducibility. High cell viability is essential. I know that was harped on a lot. Of course, we talked about the dead cell removal kit is fast and effective for increasing your viability. We also talked about isolation of target cells with our magnetic beads, as well as our Taito system, Taito cell sorter system. And again, ending on the MaxQuant analyzer, giving your reliable cell counts. And I appreciate you guys paying attention. Thank you for your attention. I know there was a lot of material thrown at you there. We hope you guys walked away with some tips and tricks. There may be some new ideas. Feel free to type any additional questions in the chat. I will stop recording and then we can take some questions.